Genetics is an ever-expanding field where scientists are constantly making new breakthroughs. One of the most exciting new realms is genetic engineering. Genetic engineering means using technology to arrange genes for purposes that are helpful to humans. When humans rearrange DNA from two or more different organisms, it is called recombinant DNA. Recombinant DNA does not occur naturally, but can be useful in many ways. Let's examine some of the ways that genetic engineering has benefited humans. If you've ever been to the doctor for a checkup, chances are you've had to get some shots. While no one really likes injections, oftentimes they are vaccines that help to keep us healthy. Vaccines are intended to stimulate the immune system so your body can recognize and fight off a harmful virus if you're exposed. Vaccines used to be made of weakened or altered strains of the harmful virus. Injecting the weakened virus allows the body to create antibodies, so it becomes immune to the disease. However, it leaves patients susceptible to actually contracting the illness and experiencing symptoms. Now it is becoming common to genetically engineer vaccines in order to reduce the risk of illness. Scientists insert genes from the bad virus into the genome of a harmless virus. The genetically engineered virus is still harmless, but will possess the surface proteins that stimulate the immune system against the pathogenic virus. In this way, your body's immune system can become familiar with the surface proteins of the harmful virus without actually being exposed to it and running the risk of getting sick. Your body can then recognize and quickly attack the harmful virus if you happen to be exposed later on. In this way, genetic engineering provides the same benefits of immunity with less risk of illness. Brilliant! Another useful application of genetic engineering is gene cloning. Gene cloning techniques allow scientists to make many copies of genes that code for useful proteins. Some of these proteins are used in the field of medicine. So-called therapeutic proteins, such as hormones, antibodies, and growth factors, are used to treat people with various illnesses. For example, the hormone insulin is needed by diabetics in order to control levels of sugar in the blood. Individuals with diabetes used to be treated with insulin harvested from cows and pigs. Today, human insulin is mass-produced by bacteria that have been genetically engineered to contain the human gene for insulin. Now diabetics can be treated with human insulin instead of insulin from cows and pigs. In addition to therapeutic proteins, gene cloning techniques have also been used to help make biofuels that are used to produce energy. For example, ethanol is an energy-rich molecule produced by fermenting the sugars in crops like corn. Ethanol is mixed with the gasoline used to power our cars. Most gas stations have signs that tell you how much ethanol is in the fuel. Mixing biofuel with the gasoline helps to conserve limited fossil fuels and can also help to reduce carbon emissions that contribute to global climate change. Scientists are currently working on genetically engineering microorganisms that can increase ethanol production using less crop material with lower carbon emissions. In this case, genetic engineering could increase our fuel supply and decrease carbon emissions. Sounds like a win-win. Let's take a look at the specific steps used in gene cloning. First, restriction enzymes are used to cut out the gene of interest, for instance, the human gene for insulin. Restriction enzymes are also used to cut out bacterial plasmids. Plasmids are circular pieces of DNA that are separate from the main bacterial DNA. The restriction enzymes cut the DNA so it is jagged at the ends. These jagged edges are called sticky ends. After cutting the DNA with restriction enzymes, the gene of interest is mixed with the plasmids. The sticky ends of the DNA stick together, combining the gene of interest with the plasmids. A second enzyme called DNA ligase is used like glue to permanently bond the DNA fragments together. The new plasmids are called recombinant plasmids. The new recombinant plasmids are inserted back into the bacteria. The bacteria that carry the new gene are called the vector for the gene. As the vector bacteria reproduce by binary fission, the gene of interest gets copied along with the rest of the bacterial DNA. Gene cloning is occurring as many copies of the gene are made. After numerous copies of the gene have been cloned, the processes of transcription and translation conducted by the vector bacteria can mass produce the gene product, for example, human insulin. As you can see, it's pretty amazing what can be accomplished using genetically engineered bacteria. Sometimes, however, the gene products that scientists want to clone are too long or complex for bacteria to produce. In that case, scientists have used mammals like cows to produce the more complex proteins. Animals that have been genetically engineered to possess genes from other organisms are known as transgenic animals. One example of the use of transgenic animals is adding human genes to cows so they produce milk containing human proteins. The protein could be a blood clotting factor used to treat a person with hemophilia, a blood disorder. In other cases, the human genes introduced into the cows simply make the cow produce milk that is more like human breast milk. This genetically engineered milk could help to improve nutrition for babies who are orphaned or whose mothers cannot produce enough milk of their own. Speaking of nutrition, genetic engineering has also been applied agriculturally to improve the food supply available to humans and their livestock. Prior to this technology, farmers would use selective breeding to improve their crops and livestock. They would select the plants and animals with the best qualities and breed them to create the next generation. Selective breeding requires a lot of time to perfect the crops and herds, however. Genetic engineering, on the other hand, provides an accurate and fast way to introduce the perfect genes into the target plants and animals. Oftentimes, those organisms are called GMOs, or genetically modified organisms. One example of a GMO is Roundup Ready soybeans. These soybean plants have been genetically engineered to be unharmed by the herbicide Roundup. Farmers can therefore spray their whole fields with the pesticide and kill the weeds, but not their soybean crops. Another example of a genetically engineered crop is golden rice, which contains one gene from corn and another from a bacterium, so it produces vitamin A, a nutrient necessary for good health. Many individuals in developing countries have diets lacking in vitamin A. These individuals suffer from preventable blindness and immune system deficiencies. Golden rice was developed in order to provide a readily accessible source of vitamin A to people who might not otherwise receive vitamin A in their diet. In this case, genetic engineering has made food more nutritious to prevent malnutrition in developing countries. Still, other plants have been genetically modified to repel insects, so farmers don't need to apply as many pesticides on their crops. This could have environmental benefits since pesticides have been documented to contaminate bodies of water and even to harm bird populations. Based on what we've discussed so far, it sounds like genetic engineering is nothing short of fabulous. But not everyone is convinced of its merits. Many individuals are concerned about the potential drawbacks involved in genetically engineering organisms and the possible impacts on human health and the environment. Opponents of genetic engineering fear that scientists could accidentally create new viruses or bacteria that might result in severe illnesses in humans, livestock, or crops. Another concern is that food containing GMO material might trigger new allergies in the people consuming them. The truth is, no scientific studies have been conducted to figure out whether there are any long-term health effects to consuming genetically modified foods. The technology just hasn't been around long enough to know if we're slowly poisoning ourselves with GM foods or not. Much of the foods we buy in grocery stores already contain GMOs, but people are unaware because labeling foods that contain GMOs is not required in most places. Certain groups are lobbying the government to pass laws that require GMO labeling on food. Then consumers would easily be able to tell which foods contain GMOs and avoid them if they felt alarmed. So far, the U.S. government has not passed any laws requiring nationwide GMO labeling, though some individual states have. Do you think consumers should be aware of which foods contain GMOs? There is also fear that genetically engineered traits could accidentally end up in wild populations. Then we might see weeds that are completely resistant to the herbicides we use to kill them, insects that are unaffected by insecticides, or bacteria that are unharmed by antibiotics. There is no telling how these genetically modified organisms could impact the environment if they were accidentally released into wild populations. Food chains could be disrupted, or delicate ecosystem interactions could crumble and fall apart. There is no way to predict how far-reaching the impacts could be. Beyond the concerns for the environment and human health are questions of ethics. Many transgenic animals seem to have poor health and a decreased life expectancy. Some die of unknown causes. Do humans have a responsibility for the well-being of these animals, and is it ethical to experiment with their genes? Another question lies in the ethics of cloning. We currently possess the knowledge and technology to clone animals, including humans. Animals like sheep have already been cloned, and that could lead to someday cloning humans. What do you think? Is it okay to clone humans? Should scientists be allowed to genetically engineer humans with traits that are considered good? These are tough questions that society will have to answer, and sooner rather than later. Now that you've heard the pros and cons of genetic engineering, what is your opinion? How far should we take this technology? See if you can strike up a conversation with a friend or relative about GMOs. Maybe you could earn extra credit for conducting a survey or interview about this controversial and very interesting issue.